All right, so I am an idiot and forgot to record our Zoom meeting yesterday, so I'm going to go over it today for everyone. This one is on DNA structure and DNA replication. We've already gone over this stuff in class right at the end of our time together, but I feel like it's been long enough that it's worth reviewing. So here it goes. First and foremost, remember that DNA, RNA, and ATP are all nucleic acids. Nucleic acids are one of the four major macromolecules. The other three are proteins, lipids, and carbohydrates. All of these macromolecules are polymers. All polymers are made of individual monomers that are linked together. The monomer of a protein is an amino acid. The monomer of a lipid is a little bit weird. We generally call them either fatty acids or steroids. We'll go into that when we do our review of biochemistry. The monomer of a carbohydrate is a monosaccharide. It's really easy to remember the difference between a monomer and a polymer for carbohydrates because the monomer is a monosaccharide and the polymer is a polysaccharide. Now, the monomer of a nucleic acid is a nucleotide. Nucleotides are made up of three different pieces: a sugar, a phosphate, and a nitrogenous base. We name these monomers based on the nitrogenous base they are attached to, hence adenine, guanine, cytosine, thymine, or uracil. The sugar for nucleotides is always a 5-carbon sugar. In RNA, that sugar is called ribose, and in DNA, it is deoxyribose. That's where the D and the R come from in DNA and RNA. The shape of these sugars becomes really important a little bit later on when we talk about the total structure of a DNA molecule. Now, the phosphate is always going to be negatively charged. When we pull up a diagram of phosphate here in just a minute, you can see that there are a ton of negatively charged oxygen molecules. This seems really stupid and trivial, but it becomes really, really important when we start talking about biotechnology and you start doing some of the virtual labs. And you can see this phosphate group right down here and all of those negative charges. So, taking a look back up here, our nitrogenous bases are divided into two different groups: purines, which are nitrogen-based molecules with 2 carbon-nitrogen rings, and pyrimidines, which are made of just 1 carbon-nitrogen ring. The purines are going to be adenine and guanine. The pyrimidines are cytosine and thymine in DNA, and uracil in RNA. So that means that the bases that make up DNA are adenine, guanine, cytosine, and thymine, and the bases that make up RNA are adenine, guanine, cytosine, and uracil. Thymine only exists in the DNA language, and uracil only exists in the RNA language. Now, let's take a closer look at these sugar molecules. In organic chemistry, everything is based in carbon, especially carbon rings. So we need a way to talk about which carbon things are attached to. The best way we've come up with is to number these carbons in a carbon ring based on where they lie in the carbon ring. We start numbering them to the right of the most electronegative molecule in the ring. This is the oxygen here, but you don't really need to worry about that for the purposes of this class. So right here is our most electronegative molecule. So as we go around here, remember, this is our oxygen. So we have carbon 1, carbon 2, carbon 3, which becomes important later on to actually make that look like a 3, carbon 3, carbon 4, and then way up here is going to be carbon 5. You can't really see it on this molecule that I'm drawing on right now, but if we were to look down here, we can see that this one right here is carbon 5. Carbon 3 down here and carbon 5 up here are the two most important carbons in this molecule. So we can see this is really where the 5' and 3' thing comes into play here. 5' is talking about the end of the sugar molecule that has the 5 carbon. 3' is talking about the end of the sugar molecule that ends with the 3'. So as you can see, we have two strands of DNA molecules that are running in opposite directions. So up here, we have our 5' carbon, and over here, we have our 3' carbon. We're able to discuss the sides of DNA by labeling them the 3' and 5' ends of a DNA strand. We can see this based on which carbon is at the quote-unquote end of the strand. So this is the 5' side, this is the 3' side. Focusing specifically on DNA here, we'll come back to RNA later, we can look at some of the structure. Now, DNA is double-stranded, which is why we say DNA is a quote-unquote double helix. The two strands are anti-parallel. If we think back to our image from the last slide, we can remember that the top of one strand was 3' and the top of the other side was 5'. This means that if one strand is running one direction, the other one is running the opposite direction. Think of two lanes on a two-way highway. One side runs north and the other one runs south. The backbone, or sides, of the two DNA strands are alternating phosphates and sugars. They're connected together with those strong covalent bonds. Remember, you don't want your backbone to fall apart, you want it to be really strong. Having your backbone fall apart would be a very, very bad thing. This is the exact same for a DNA molecule. You don't want it to fall apart either, so we have to use those really strong covalent bonds. The nitrogenous bases run up the middle like rungs of a ladder. You will always have a purine paired up with a pyrimidine. This is because you always want 3 rings going through the middle of the molecule. Otherwise, it would get all squiggly and messed up. Thymine will always bind with adenine. In DNA, remember we're going to come back to RNA later, and guanine will always pair with cytosine, in both DNA and in RNA. As you can see, the bases have to bind together because these are the only places that hydrogens will match up. Remember, you need a hydrogen attached to an electronegative molecule and another electromolecule to form a hydrogen bond. So if we look here, we have a hydrogen bound to an electronegative nitrogen, and then an electronegative oxygen. So you can form a hydrogen bond here. Here we have a hydrogen bonded to an electronegative nitrogen and another electronegative nitrogen. So thymine and adenine are the only ones that could form these bonds in this direction and in this way. Cytosine and guanine are going to form 3 bonds. One here, and one here. Adenine and thymine are only going to bond twice. What this means is that if you have a ton of cytosines and guanines in a molecule, that DNA is going to be a lot harder to break apart. This is often written as cytosine and guanines have a higher boiling point. I don't know why this is really important, but it's just one of those random things that the AP test seems to find important. I don't know why, but it does. All right, so moving from DNA structure to DNA replication. Every time you divide a cell, you need an extra copy of DNA. That way, both of the daughter cells have a full set of DNA to function with. There were a couple of initial ideas on how replication happens, but we now know that the process is known as semi-conservative. This means that each new strand, quote-unquote, of DNA is made of half of the original strand and half new DNA molecules strung together. It's possible to replicate these two identical strands because of those base pairing rules. A pairs with T, C pairs with G. By the way, we also call these base pairing rules Chargaff's rules. Now, this process can be explained very simply. Step 1, unwind the DNA. Step 2, add in new DNA nucleotides. Step 3, fuse together the new backbones. Now, there are a bunch of enzymes that are involved here, and it makes it slightly more complicated, but that's the most basic way to understand it. DNA in eukaryotic organisms, and remember, those eukaryotic organisms are 1, plants, 2, animals, 3, fungus, and 4, protists, are huge. If you were to start at one end of the DNA strand and go to the other, like prokaryotes such as bacteria and archaea can, it would take you forever. To speed this up, eukaryotes replicate in a bunch of different places at once. We call these replication bubbles. Now, there are 5 majorly important enzymes that help this process to happen. We're going to talk about them in context here in just a minute, but we're going to talk about each of them now. First, there's helicase. This enzyme is the enzyme that unwinds the DNA in the first step of replication. Second, there's topoisomerase. This enzyme helps to relieve the stress on the DNA strands that builds up by pulling it apart. Knots don't like being pulled apart. It needs help to not just fall apart entirely. It does this by clipping one side of the DNA and then refusing it later. Next up is primase. What you have to realize is that the enzyme that adds in the new DNA bases is really, really stupid. It needs help to find where it should bind. Primase comes in and adds in that RNA primer so the DNA polymerase, coming up next, knows where to go. Then we have DNA polymerase. This enzyme has 2 major functions. First, it adds in new complementary bases, and second, it removes the RNA primer and puts in DNA bases. There are technically 2 forms of DNA polymerase, but all you need to know is that it has 2 major jobs. Finally, we have ligase, which we're going to look at here in just a minute. Now, these are more detailed steps of DNA replication with the enzymes added in. If you want, you can pause it here and record those, but they're pretty self-explanatory. Now, we can see that primase binds to the strand that starts with the 3' carbon, right here. And it lays down an RNA primer. DNA polymerase then binds and lays down the actual DNA bases so that the DNA strand is built starting with 5' and ending with 3'. However, the strand that it's being built off of, which in this case is called the leading strand, more on that later, is 3' to 5'. This is always something that I struggled with, so make sure to take extra time and attention to look at it. However, this is when we can remember that DNA polymerase is really, really stupid. It can only add bases to the 3' carbon of a nucleotide. However, DNA is anti-parallel, which means that only one side of the DNA molecule can be replicated all at once. This is the leading strand. The leading strand starts with the 3' carbon. The other side has to be made of small sections called Okazaki fragments. RNA polymerase adds in a bunch of RNA primers as helicase opens the strand up, and then DNA polymerase adds in the DNA bases a couple at a time. This is where finally our enzyme ligase comes into play. Ligase is responsible for stringing together these Okazaki fragments. As we can see here, the leading strand is made all at once. The new DNA strand is made 5' to 3'. The lagging strand, on the other hand, is made into small sections, with primase adding in a bunch of RNA primers, one here, and there was one here. And DNA polymerase creates those Okazaki fragments, and ligase, here labeled DNA ligase, is stringing them all together in fragments. So that's all I have for nucleic acid structure and replication. Next up is going to be protein synthesis, which is explaining why we need DNA.